So first, thank you to Polina for giving us the opportunity to talk here. And I think we've heard a little bit about the 100,000 genomes and the DDD study, and we're slightly different. We're maybe slightly more focused. And those of you who have received the SWAN newsletter back in October will see that we had a section here about what we do in our group. So we thought it might be a good opportunity to maybe give a bit more detail about how we work as a group. So yeah, so we work in the Institute of Child Health. And just to give you a bit of background of who we actually are, well, we get our funding in quite a unique way. So we're part of UCL, but we also gain our funding through the BRC, which is linked to Great Ormond Street Hospital. So we kind of have this this crossover between the clinical side and the research side, and that puts us in quite a unique opportunity and a position to do the sort of work that we do. And really the idea of setting this group up was, it was set up by Professor Phil Beals back in February 2010, was really to bring the expertise that we have in terms of, as you've been hearing about next generation sequencing and the interpretation and the analysis of the sort of genetic work, and really to bring that to the clinical professionals who work in Great Ormond Street. This is such a fast-moving area of genetic research that you really need to be quite an expert in it. And obviously, people who are working in the hospital maybe not have the expertise, but we, by marrying the two groups together, we are able to offer them our expertise, and they're obviously able to give us the samples that we need to analyse. And as the overarching theme that we have is to facilitate this rapid gene identification. And the reason why it's pertinent to come to a meeting such as this is because we look for the genes that predispose to rare, uncharacterised genetic diseases, such as those characterised by the SWAN. And really, our main idea is that by finding by identifying the genetic mutations in the patients, we can lead to a better diagnosis, which leads obviously to better clinical management. But really, what we're able to do and what we aim to do is by finding the genetic mutation, we're able then to go maybe a step further and really to look at the biology of that. So we look at the functional studies to try and determine why that genetic mutation causes the phenotype that we see displayed in the patients that we're looking at. And also this leads on to translational research. And really this is taking it from just looking at the gene to looking at the biology to actually seeing how we can move that forward in terms of allowing us to have better diagnostic testing. If we find the identified genes for uncharacterised disorders, we then got obviously a new gene that we can look for in patients with similar phenotypes, so we can improve their diagnosis. This obviously leads to better genetic counselling for those families. Obviously a big part of this can be prenatal service, and obviously the end point is by finding a mutation, by trying to by elucidate the biological mechanism that's leading to this phenotype, we're able to better provide maybe therapeutics to be able to treat these disorders in the future. So really, what do we do as a group ourselves? We're a bit of a one-stop shop when it comes to the analysis and the gene identification side. So we work with the clinicians, and our first port of call, we will actually we will look at the evidence for the inheritance patterns in the families to see whether it is possibly a genetic disorder. We will arrange the sample collection from the clinicians. So we go directly from the DNA. We then perform all the QC and prepare the DNA ready for sequencing. We send the DNAs away to specific sequence providers who actually do the sequencing for us. We're expanding this to include more in-house UCL services as well. But once the sequence providers have done their sequencing, and we normally look for exome sequencing, so although we're again, as many groups are, we're looking to go forward into whole genome sequencing, because of the interpretation problems associated with that at the present time. We're really focusing just on the exome. So we take the exome data, we then perform the analysis on the raw data. This is no trivial undertaking. This is one of the real problems with trying to deal with this kind of data, is that you end up with tens or hundreds of gigabytes worth of data. So you need a large computational infrastructure to be able to not only handle the data storage, but also to actually analyze the data. So we employ specific bioinformaticians who are able to actually analyze this data for us. And that leaves us with a list of variants. As we said, we all know that we have variants between one another, and that these variants may be causative for the disorder, or they may just be natural variation that we all share. And that's the real difficult part, is how do we interpret those findings. And so we use cutting-edge software that allows us to filter the data and hopefully bring us down to what we think is the single gene point mutation that's causing the actual disorder. And then when we get to this point, we again go back to the clinician, and it's an iterative business. We discuss with them the findings. We try and discuss the likelihood that the mutations that we find are actually causing the disorder. And then, obviously, as I said, we then try and move on translationally to find out the biological mechanism that's actually leading to the phenotype. So I thought I'd just put this up just to give you an idea of some of the phenotypes that we look at. Because of the unique position that we have linked with Great Ormond Street Hospital, we obviously see a huge number of different phenotypes coming through. So far, we've analysed samples from in excess of 60 different clinical phenotypes. Some of them may be syndromic or have syndromic features, but the majority of the ones that we try to look for are the ones that are interesting to people here for the SWAN meeting, in that they're uncharacterized, they're non-syndromic. And so, although it's exciting that you may be able to find novel causes, it's obviously more difficult to actually find what may be the gene identification side of it. So that's an idea of some of the work that we've been doing, but I thought we've only got a short amount of time, but I thought I'd just try and give you a feeling of how we fit in with this the UK strategy for rare diseases. So this was a UK-wide strategy was released last Friday. We were here last Thursday to have the unveiling of it in this very room here. So I thought it might be interesting for those who've looked at this to see where a group such as ourselves will fit in. Well, obviously we fit in with the role of research, being part of of UCL being a research-based organisation, and it's important to really make the distinction that we are not a clinical lab. So we don't do clinical diagnostic sequencing ourselves. So if you have if you're a family and you have someone who's affected and you want to be involved, we can't personally do that ourselves. But what we can do is that you could go to your clinician and they could refer you to us as a group. And if you fill our criteria for inclusion in the study, then we can enroll you into our group and we can perform exome sequencing. But we are not the first point of call. So I really want to make that quite clear. We're still very much based in research and not in clinical diagnostic. But it's also important to sort of think about how our role is expanding. So I think it's when we find mutations that we think are causative for the specific genetic disorders that we're looking at, it's important that these can then lead to diagnosis. So as I said, if we find a gene, then we work very closely with the clinical labs here, so we can make a clinical diagnosis when we find something. And obviously this is helpful in terms of leading to early interventions. And also because, and this is where the theme of translational comes in, we're working as a group, we're working with people who work in more of what we call proteinomics, so working with the proteins in the cell. And really we're looking at trying to find biomarkers that people can test that may show up a gene deficit in something. Instead of having to give a DNA sample, maybe you can just give a urine sample or something. And this will allow for better screening, and by better screening we'll be able to identify the sort of disorders that we look at. We'll be able to identify them earlier. By identifying them earlier, we have the opportunity to treat them early, and by treating them early, we have a better opportunity to either alleviate the symptoms or prevent the symptoms from starting in the first place. And I think, yeah, so I'd just like to end there, and just to really show that as a group, the GOSgene team, we work with collaborators from across the UCL network, and obviously with Great Ormond Street Hospital. I think we'll...
Just one final question, which, you know, if I'm the parent of a child without a diagnosis, and you know my doctor thinks you might be able to help with that, so he sends a sample to you. Is there a fee? Is it a free service? How does it get? How do patients get into the service? Does the hospital have to pay for them to be sequenced, or is the funding from some other source?
Well, we're in quite a unique position in our group, because our funding actually allows us to do the sequencing without cost recovery. So we are able to meet the costs of the sequencing, but by doing that, we have to then ensure that the samples and the families that we recruit are those that are most likely to give us an outcome from the cost. So there wouldn't be a cost to the hospital. We would take that on board ourselves.